A well-designed experiment gives us the answers we're looking for. A poorly designed experiment, at best, gives us nothing, and at worst, misleads us. So designing experiments well can be very important. Let's look at an example. The Physicians' Health study looked at whether aspirin and/or beta-carotene had any effect on a variety of medical conditions, including heart attacks, several forms of cancer, and others. So let's talk about some vocabulary. We will talk about experimental units or subjects. These are the individuals on which the experiment is done. People are called subjects. Everything else are called experimental units. To call people experimental units seems a little dehumanizing. We can talk about the experimental treatments, or treatment, which is the experimental condition or conditions which are applied to the subjects. Now, in this case, aspirin and beta-carotene are the treatments. Factors are really just another word for variables, usually the explanatory variables. In this case, again, the treatments and factors aren't too much different. Factors are aspirin and beta-carotene. And finally, we can talk about the number of levels. Levels are the number of different amounts used for each factor. So, for example, in this Physicians' Health study, there were 2 levels. There was the presence of aspirin or non-presence of aspirin, the placebo. You could have, 3 levels in which you could have non-presence and 10 milligrams and 20 milligrams. And that would be 3 levels of aspirin. So that's what I mean by the number of different amounts. So it's not the amount itself, it's just how many different amounts are being used. So for this example, we're actually going to use 2 factors and 2 levels. And because we have 2 factors and 2 levels, we're going to have 4 experimental groups, which can be diagrammed kind of like this. We have a group that is taking both aspirin and beta-carotene, just aspirin, just beta-carotene, and neither. Because, especially for medical applications, perhaps these 2 work well together. And so we want to really figure out all the different combinations. Now, the one combination which is of particular importance is, in fact, this one. This would be the control group, which is very important in experimental design. There's actually a really clear example of this. I'll show it to you. At one point in time, there was a medical procedure called gastric freezing. This was supposed to reduce pain in ulcer patients. And essentially, a small balloon would be put down into the stomach, and cold liquid would be circulated through, reducing the temperature, and patients said that they were feeling better afterwards. Became widely used before any real scientific study was conducted. Eventually, someone was like, well, maybe we should actually see how effective this thing is. And an experiment was produced. And we had those who felt they were improved was about 34% felt improvement when they had gastric freezing. And there was a control group, of course. 38% of the control group felt improvement. Which is known as the placebo effect. Really, the gastric freezing clearly didn't have much to do with it. More people felt improvement with the placebo than the actual treatment. This is a pretty powerful effect, I've been told. When people feel they are being treated, they get better. It might be because, for whatever the reason, they take better care of themselves, because people are checking in on them. There's a lot of different psychological factors that go into that. But the effect is real, and this actually demonstrates it pretty nicely. So without a control group, you can create and produce statistics and say, so many people felt improvement, or this was the rate of heart attacks or cancer. But unless we have a control group to compare it against, it doesn't really tell us as much as we want. Because what this really comes down to, again, it's a common theme, working variables. If we want to try to eliminate the effect of working variables, then we need a control group. Because whatever working variables are in the 3 experimental groups should also be in the control group. So if there's some systematic working variable, at least everybody has it. And we can compare against the control group and see what effect the aspirin and the beta-carotene have. That's really what it comes down to. The next important issue to try to reduce working variables is a design element called, and that's not the correct way to begin, it's double-blind, not blind-double, the double-blind design. Just like in surveys, remember that the attitude of the interviewer could have quite an effect on the outcome of the survey? Well, if the experimenter knows a certain person is in the control group and it's a placebo, there are a lot of cues that experimenter can give, a lot of body language, word choices, all kinds of subtle things which the subject doesn't even realize, but is still affecting them. So double-blind means neither subject nor experimenter knows who is in what group. That way, they can't be unduly affected, or even unintentionally affected. Now, I do have to qualify a little bit what we mean by the experimenter. Someone in the experiment probably needs to know who's in what group. But that person shouldn't have direct and consistent contact with the subjects. The experimenter who is actually handing out the pills, that experimenter needs to be blind, needs to not know who is in what group. The head researcher, that person can know, because that's not going to affect the subject. Now, what's also going to be important is that every group needs to have the same treatment, right? Which is to say, you're going to have 2 pills. One's going to be a white pill and one's going to be a red pill or a capsule or whatever it is, but everyone's the same. The placebo has to look and feel and taste as much as we can the same as the real thing. That way no one can tell who has been given a placebo and who has not. So because this was a real study, it was actually quite a large study, almost 22,000 subjects, all of them male physicians. That's why it was called the Physicians' Health Study, because the subjects were physicians. And they're all male, which of course means whatever conclusions are drawn really only apply to males. If we wanted to include females in the conclusion, we really needed to have females in the study. Some of the results, they of course looked at a number and variety of medical conditions. Only one of them seemed to really come out as significant. The control group suffered 239 heart attacks. The aspirin-only group, 139 heart attacks, 100 less, which is 42% fewer heart attacks of the people taking only aspirin versus the control group. That seems fairly significant. There is, however, an important question. From our understanding of statistics up to this point, we know there's variation. So we would actually expect variation between these 2 groups. We wouldn't expect them to have the same number. So a difference of 100, which is 42%, Is that typical for the variation, or would that be really unusual due to just random variation? That actually is the essence of inferential statistics right there. And we're going to, once we finish talking about designing experiments, we're going to figure out how to actually draw those conclusions of significance. Is this significantly different, or is it just different because of normal variation? We can take what we've been talking about here and summarize principles of experimental design in 3 basic principles: control, randomize, and replicate. We've talked about the first 2, really. You need to have a control group. That way, whatever lurking and confounding variables there are, they're in the control group also, so it kind of comes out in the wash. We need randomization. Right? The question which I always ask when students are working on this stuff, if you're designing an experiment, where is the randomization? In this one, randomly divided into 4 groups. That's the randomization. Again, if there are lurking or confounding variables, hopefully by doing this randomly into 4 groups, whatever lurking and confounding variables there are, there'll be the same amount in all the groups, and again, it kind of comes out in the wash. Replicate, we haven't really talked about too much. This is essentially having enough subjects or units for a good-sized sample. Right? If this study didn't have 22,000 subjects, but there was like only 2 people in each group, would we be as sure about our results? No, not really, because with very small samples, just intuitively you would think there's probably going to be just a lot of variation, because one small lurking variable is going to really throw things off. But with 22,000 split into 4 groups, that's going to really, it's the law of large numbers from probability. We want a good-sized sample. We're going to talk, when we begin inferential statistics, like what does it mean to be a good-sized sample? That gets, fairly technical, and we'll talk, actually we talked a little bit about t-distributions, which are for samples, and that, as the degrees of freedom, which is the sample size, goes up, it starts approaching the normal distribution. Well, that's going to guide us as to what's a good size. For the moment, I'm just going to leave it a little bit general as just a principle of good experimental design. An interesting question sometimes comes up at this point when we're talking about experimental design. What about physics experiments? Or physical experiments of some kind? Well, we definitely want to, let's work on a specific example perhaps. How about we want to experiment and see what the strength of a steel beam is, right? So what are we going to do? We're going to grab probably a bunch of steel beams, put them in a testing machine, and test them until they break. Well, we certainly have replication because we want to do that with more than one steel beam, right? That would be good design. Randomize, we want to randomly select the steel beams. That's not usually a big deal. Someone just goes out into the yard and grabs them, and then it's pretty much randomized. The real question comes into, what about control group? Physics experiments don't seem to have control groups, the way the physician's health experiment had a control group. Well, it turns out that really the purpose of a control group, as we've talked about, is to try and minimize lurking variables. Right? That's the purpose. Well, the lab does that for physics experiments. That's the whole purpose of doing a testing in a nice controlled laboratory. If we were to try and test the strength of a steel beam after it's been put into a building, for example, even if the contractor would let us do that, then how do we know if the fail strength or fail force is due to the steel beam, or maybe the connections, or there's all kinds of factors that go into the real world out there. So the lab actually is the control for a physics experiment. Now, experiments are great, and they can tell us a whole lot, and we've been very successful with experiments. That's why we now have computers and iPads and iPods, and it's all the result of experiments. It's very cool stuff. But there are some cautions that we need to actually keep in mind when we experiment. The first caution is lack of realism. And again, this is usually more involved with psychology or sociology experiments. The physics experiments, that's okay. The lack of realism doesn't really matter so much. One example is a study comparing two commercials. Right? We have a researcher that gets a bunch of kids. He wants to know which commercial they like better. Brings them into a lab. They watch it. They say, I like the first commercial better. There's a certain lack of realism in that situation. If they were able to actually view the commercials in their living room with their friends around them, not necessarily knowing that they're being studied, maybe they would respond differently. It's actually a little bit hard to know. But there's actually an ethical issue here. Right? Well, ethics. People need to know if they are an experimental subject. It's different than an observational study. People can be observed, and that's not much of an ethical issue, more or less. But if you're applying a treatment, some kind of experimental condition on someone, they need to know, and they need to accept what they're being subjected to. So that gets a little tricky sometimes. Another really good example, I love this one, is rear-end car collisions when you have two taillights, or whether there's a third one that's usually kind of center rear windshield. There was a study, right, and they found that with the third taillight, there was a drop of 50% of rear-end collisions. Well, that seems quite significant. So it was actually mandated. After a certain time, I think it was 1986, that there had to be a third taillight in all manufactured cars. Sometime later, they did a study to see about the rear-end collisions, and it was down about 5%. Huge difference. What was the difference between the study and after it was mandatory and it was on the roads? Well, the thought is that during the study, very few people had three taillights. So those who did, that were part of the study, other people noticed it right away, and there was a reduction in collisions. After everybody had taillights, it wasn't that noticeable, it wasn't different, so people stopped paying attention, and there wasn't as noticeable a decrease. So the study, there was a lack of realism because there weren't many cars that have three taillights, which was what the real situation was going to be in.